All right. Hello, everyone. Okay. Hello, everyone. So today oh, why do I have that slide? One second. I'm not projecting the slide I want. Okay. All right. Hello, everyone. Welcome back. Today we're gonna do lab 4, chemistry, biochemistry, and we're gonna start digestive system. Okay. I don't know if you noticed. We were supposed to do lab 3 today because we did lab 1 and lab 2, but I've swapped lab 3 and lab 4 because last week in lecture we had chapter 2 in lecture that was a slide deck that was not a recording. So today in lab I go over this material, and then if you noticed your chapter 2 extra credit handout, if you haven't turned it in yet, I've extended the deadlines for week 1 and week 2. It's due this Friday midnight. I'll probably extend it one more time till Monday midnight, but Monday is the census date, so I won't be able to add any more new students after that, but I have been adding new students. So if you see me extending the deadlines for everyone is because I just want to make sure everyone gets their required material for the course, has access to Canvas. And then if someone was added, like, for example, 2 days ago, they won't make it to turn everything in. And so this is how it's going to be the first couple of weeks. But after next week, like week 4, because I'm not going to be adding any more students, everyone should be caught up because 2 weeks from now, okay, no, 3 weeks from now, we have our first lab exam coming up, okay? So, and then to do your best on the exam, we have to make sure that you've completed the assignments, right? Because remember, the exam is, we don't have a cumulative final, right? So everything that we learn, we have to apply it on this exam, right? I mean, we can make up some of the assignments after the exam, but it won't help for the exam, but you will get your points, I guess. But it's better to finish everything before the exam, okay? So if you've been in the class since day 1, hopefully you are caught up, okay? The extensions are not really for you, right? But I have been extending it. So if you need it, please take advantage of that. So back to what I was saying, chapter 2 is on chemistry and biochemistry. And then today in lab, I'm gonna spend some time on that. Which is part of lab 4, and we're gonna start digestive system, okay? The only thing that we have left to talk about before we just dive into all the body systems is gonna be the tissues, okay? Which we're gonna cover next week. But next week with the tissues, we're already gonna start with the next body system, which is the integumentary system, okay? And then this week, this Friday, when the next week's module is published, I'm gonna go ahead and publish the module for the week after. Okay, you don't have to do anything, but I'm gonna make it available because next week when we do the lab 3, the tissues, then after that we have lab 5, and lab 5 is part of the lab exam 1, and lab 5 is on bones. Okay, the bones are gonna be part of our lab exam 1, and I don't want to cover lab 5 1 week before the exam, and then in a week you have your exam and then you're tested on the bones, right? So I'm gonna make it available sooner. Next week in class, I'm gonna bring the handouts for lab 5. If we finish early, we can even get started on the lab 5 so that you'll have time to start studying, okay? The bones are not as hard as the muscles, okay? The majority of the bones you already know from Bio 5. And then some of the bones, we just know the names because we know it, right? And then I promise when we do go over the slides in class, like each bone, we see it in like many different views that you will be able to learn, okay? I'm not saying there are no new bones. There are some new bones that we have to learn, but we'll do our best to learn in class, and then you can review it and then be ready for the exam, okay? So I'm just letting you know, not next week, the week after, we have the bones, but from this weekend, I will make the bones available for you just so that you can start reviewing and practicing, okay? All right. So today, let's start lab 4, chemistry, biochemistry, and the digestive system. So, all right. So everyone, here we can see the structure of an atom. Okay, we have our periodic table here. In the periodic table, we can see all the atoms, right? And then with their atomic number in the periodic table. And then the atomic number in the periodic table for each atom is important because we're gonna do some things and we have to know those numbers, right? So, but let's talk about atoms first. So this is, for example, carbon, okay? And in the middle, you can see the nucleus, and then you can also see positive signs and then neutral signs, right? So the positive signs are the protons, right? And then we also have neutrons, right? And around the nucleus, we have this shell, this outer shell, right? And then on this orbit, on this outer shell, right, we have electrons, okay? So, and then if you look at the numbers, right, we have equal number of protons, which is equal to the number of neutrons and equal to the number of electrons. Do you see that? We have, for carbon, for example, we have 6 protons, 6 neutrons, and 6 electrons, okay? Inside the nucleus, we're going to have the protons and the neutrons. And on the outer shell, around the orbit, we're going to have the electrons, okay? Now, what is the charge of the protons? Positively charged. Neutrons are neutral, okay? And the electrons are negatively charged, okay? And then the electrons are found on the orbit, on the outer shells, and then there's some rules that these electrons need to follow on the outer shell, okay? So that the atom is stable, okay? So as you can see, carbon is carbon 6. It means it has 6 electrons, okay? And then if we look at the first orbit, first outer shell, how many electrons do we have there? We have 2 electrons, okay? And the reason why, even though we need to place all 6 electrons, we only have 2 because the first shell, right, it follows the duet rule. The duet rule means that we can have up to 2 electrons. And then if we have 2 electrons, it means it's full and it's stable, okay? Now, carbon is 6, okay? We have 2. How many do we have left? 4, right? We still have 4 left. So to add the 4 electrons, we're going to draw a second outer shell, right? So here we draw the second one, and then we start placing the next 4 here. So the second shell, everyone, it follows the octet rule. Octet means 8. So for us to make this atom stable, right, we have to satisfy the octet rule, which means that it needs up to 8 electrons, okay? So here, how many do we have on the second one? We have 4, right? So to make it stable, we can either add 4 more, right? And then it will be 8, or we can donate these 4 to another atom, right? And then we lose the second outer shell. We'll have only the first one, but the first one is already stable because it has completed the duet rule, right? So just wanted to go over this before what we have to cover next um, about the structure of a representative atom. And then everyone here, this is the periodic table. Usually this is given when needed because from the periodic table, you can get the atomic number, which is the number that we need to be able to draw all these electrons that we've been talking about, okay? So the ones that I want you to look at right now, because we're going to do it in the next 2 slides, is sodium and chlorine, okay? How do you write sodium? Na, right? What's the atomic number for sodium? Na11, right? And then the next one is chlorine, right? What's the atomic number for chlorine? 17, okay? So let's just keep in mind these 2 numbers, okay? One is Na11, the other one is chlorine 17. First, we're gonna start with the sodium one, okay? So for the sodium, I'm gonna go ahead and I want everyone to like just open a piece of paper, right? I'm gonna draw here because I want I'm recording and I want my voice to be loud. And I'm gonna share the recording with you. We're just gonna draw here, but I want you to draw with me and follow along, okay? So everyone, we said that we have sodium. What's the sodium number? 11, right? Mm. Sodium 11, and I'm going to write Na in the middle, Na11, right? And then please help me place these electron shells around the sodium, right? So I'm gonna do the first one, right? And then how many electrons can I add on the first shell? 2, 1 and 2. Okay, so we need 11. Okay, so I'm gonna have the second one because we completed the duet rule. 3, 4, 5, 6, 7, 8, 9, 10. Okay, I'm at 10 electrons and I already completed the octet rule on the second one. So what do I do? A third one, right? And then the third one, everyone, also follows the octet rule, okay? So I'm gonna draw the third one and then I'm gonna put the electron here, okay? So now, just like we talked about it with the carbon, right? Is the sodium here like this? Is it stable? It's not stable, right? Because on the outermost shell, right, it has 1 electron, right? It does not have 8 electrons total, right? So there's 2 things we can do. We can either add 7 more electrons, like, and we need another atom to donate to us 7 more, or we can donate the 1, right? Okay, so that if we donate the 1, then we can get rid of the outer shell, then the second one will be the last one, and the second one follows the octet rule, which means it's stable. Okay, so whenever we see ourselves in these situations, and for example, here you have 1 electron by itself, and you don't have a full outer shell, we say that here for this one, we have 1 valence electron. Okay, sodium has what? 1 valence electrons. Okay, so this is better. 1 valence electron. All right. Now, everyone, is it clear with the sodium? Can we do the chlorine next? Yes? Okay. So what did we say the atomic number for chlorine is? 17, okay? For chlorine, it's 17. So Cl17. So I'm going to put here Cl17 in the middle. I'm going to draw the first circle. So in the first one, how many electrons can I put there? 2, and then we're done, right? And then I'm going to add the second one. 3, 4, 5, 6, 7, 8, 9, 10. I completed the second one. What do I need? Another one, right? So I'm at 10, 11, 12, 13, 14, 15, 16, and 17. Okay? So here now on my outer the outermost shell, right? What's my valence electron? 7, right? So I have the valence electron for chlorine is 7. We have 7 valence electrons. Okay, we have 7 valence electrons because we don't have a stable atom because it's not following the octet rule. So we can either get rid of the 7 electrons and then it will be stable, or we can have another atom donate one of its electrons. So here we have sodium that needs to get rid of 1, right, to be stable. And then we have chlorine that needs 1 electron to become stable. Okay, so this is where sodium is going to donate that 1 electron to to chlorine, okay? And so that now sodium will be stable and chlorine can be stable. Now, whenever this happens, whenever we're donating electrons or accepting electrons, okay, what happens is that sodium, sodium is donating electrons. It means that it's getting rid of an electron. And then what is the charge of electron? Negatively charged. So if you're giving away negative, you're becoming less negative, if that makes sense, right? So when you're becoming less negative, it means you're becoming more positive. So whenever sodium donates an electron to chlorine, it becomes Na plus. It becomes positively charged because it's becoming less negative because we're getting rid of an electron, okay? And then chlorine is accepting, receiving an electron. And then we said electron is negatively charged. So chlorine is becoming more what? More negative. So that's why chlorine gets the Cl minus, right? So whenever you have Na plus and Cl minus, right? Sodium chloride, this is an ion, right? And then um, the type of bond that's happening here between these two atoms is what? So what are the two type of bonds that we have? I know I'm going ahead, but we'll get to that slide. What are the two types of bonds that we have? Ionic bond and covalent bond, right? So in this example, is this an ionic bond example or a covalent bond example? This is an ionic bond example. It's okay, I didn't hear who said, I heard both words. It is an ionic bond example. So ionic bonds, everyone, if you are taking notes, note that whenever we are donating an electron or accepting an electron, that means we have an ionic bond, okay? Ionic bond is you're completely donating it, getting rid of it, or you are accepting a whole one for yourself, okay? So that is ionic bond, and I want you to understand what is an ionic bond very well before we cover the covalent bond so that it does not get confusing, okay? And I don't want you just like to remember which one is which. I want you to understand so that you can never forget, okay? I remember just memorizing it, and I don't know until when I used to be like, oh, which one was ionic, which one was covalent, you know? If you understand it, you don't even have to try to remember it. Like you'll just know because you understood it, okay? So ionic bond is when we're completely donating or we're completely accepting and we're building an ion, okay? It's the ionic bond. So everyone, let's check on the slides if we did everything correctly. So this was for the sodium. We said sodium has one valence electron. This was for the chlorine. We said chlorine has 7 valence electrons. And then basically we can see here that basically here we can see that something popped up here. I'm trying to close it. Okay. Basically, we can see here that how sodium is donating its valence electron, that one valence electron to the chlorine. And then sodium is becoming stable sodium, and it's becoming positively charged because it's getting rid of an electron. And chlorine is becoming stable because it's accepting that one electron, and it's becoming negatively charged, okay? And together, right, we make, because one is positively charged, one is negatively charged, or cation and an anion, we make an ion, right, through ionic bonding, okay? This is an example of ionic bond, okay? So next. So this is what we made, okay? Do we all understand ionic bonds? So that I can go to covalent bond next. Okay. So we said ionic bond is when we're completely donating or receiving, right? So covalent bond is a little bit different. Covalent bond is that, for example, And then this is the hydrogen example. What is the atomic number for hydrogen? It's the first one. It's 1, right? So hydrogen is 1, right? So if I'm going to draw hydrogen, right? For hydrogen, it's 1. I'm only going to draw 1 electron, right? Does this follow the duet rule? It doesn't follow the duet rule. What does it need? It either needs another electron, right? Or we need to get rid of this. But we don't even have another outer shell before that, right? So for example, for covalent bonds, right? Uh, let's say in this example, we have two hydrogens, right? And then both hydrogens have what? One electron and one electron, but they need one more electron to complete their uh, outer shell to become stable, to follow the duet rule, right? So in covalent bond, right, we don't see one atom donating a complete electron to the other atom or the other atom receiving electron from the other one. In covalent bond, what it happens is I need one and like I need another one, right? And then you need another one. I only have one. You only have one. Let's hold hands and share our ones so that we can both have two. Does it make sense? All right? So in covalent bond, they atoms share electrons. What do they do? Share electrons. Okay, so that's the difference. Ionic, we're donating or receiving. In covalent, we are doing what? Sharing, right? Because just like the hydrogen example, right? One hydrogen has one, the other hydrogen has one, but each hydrogen needs two, right? So if they share each one's ones, right, they will each have two, but by sharing, okay? So this is what we're seeing here in this slide. So each hydrogen atom has a single electron. Each proton attracts the electron to the other atom, and then the shells merge, and now each atom obeys the duet rule. So what type of bond is this? Covalent. And then what happens in covalent? They share electrons. No charge. No charge. And then that's the difference between ions. Ions, they get charged. Covalent bonds, there's no charge. But we're going to talk next about polar and non-polar covalent bond. That's coming up.
Bonds only happen to stabilize atoms?
Yes, yes, yes. Both ionic bonds and covalent bonds, they're doing that. They're coming together so that they can stabilize each other. Yeah. This is, any other questions? Okay, good. So everyone, this is a table. It's showing us electron sharing in covalent bonds. We said that in covalent bonds, we have sharing. And then you've probably seen this in chemistry. And then we're not going to go into details, right? We're probably familiar with single bond, double bonds, right? And then basically, single bonds is what we're seeing right now with the hydrogens. That's a single bond. Hydrogen to hydrogen is a single bond. For example, the second one, it's an example of O2, oxygen, two oxygen molecules. Two oxygen molecules, they share two electrons together because they need four in total, two of the oxygens. And then we'll see it in the example on the next slide. Okay, so if you are sharing two, then that's how many, what's that bond? Double bond, right? Or if you're sharing three, then that becomes a triple bond, okay? So this is where double bond, triple bond comes up, okay? All right, let's look at this example here, electron sharing in covalent bond. So everyone, do you remember carbon? What was the atomic number for carbon? It was 6, right? So 6 means the first one takes 2, right? And then the second one takes 4, and then we need 4 more, right? To become 8, okay? And then we have 4. And we have 2 oxygens. This is CO2, right? And then in this particular example, this is what we're doing. We have 2 oxygens. The oxygen's atomic number is 8, okay? So O8 means the first orbit takes 2 electrons, right? And then we have how many left on the second one? 6. So how many does the oxygen needs? 2, right? And we have 2 oxygens. Each oxygen needs 2 more electrons, right? And then in the middle, there's a carbon that needs how many? 4, right? So what is the carbon gonna do? It's gonna share 2 electrons with one of the oxygens on the right, and it's gonna share 2 electrons on one of the oxygens from the left. And then that's why we're gonna have double bonds, right? C double bond O and C double bond O on the other side, right? And then this is an example of covalent bond. And then look how the carbon and the oxygen, they're all sharing whatever electrons they have to complete their outer shell and become stable. Okay, is that clear? Okay, good. Now let's talk about charges. So before we go more in details with whatever I'm going to cover next with covalent bonds, are we all comfortable with ionic bonds versus covalent bonds, the difference? Okay. Also, did you understand it in a way that you won't forget, like you're not doing what I did as a student? Okay, good. Next, we're going to talk about when covalent bonds, we have polar and non-polar, okay? So before we go into details, difference between polar and non-polar. Polar means opposite sides, right? Like they're opposite. Non-polar means like they don't have that opposite, that opposite sides. So the first example on the top is non-polar covalent bond. Whenever we're saying non-polar covalent bond, so both for polar and non-polar covalent bond, there's sharing of electrons between atoms. The difference is that whenever it's non-polar covalent bond, whenever they're sharing electrons, they're sharing it equally, okay? This is like, I don't know if you have a sibling or someone super close to you, and then you're gonna split a sandwich, right? And then you cut it into half. You actually cut it down the middle. Like it's not like one of the pieces is bigger than the other, okay? So both people get equal amount. Non-polar covalent bond is when both atoms are sharing equal amount of the electron, okay? The polar covalent bond is they're sharing, but they're not sharing equally. One of them, one of the atoms, the electrons are hanging out more towards one of the atoms than the other atom. Okay, and then this is the example of, let's say you're sharing a sandwich with a sibling, and then your sibling cuts it in half, but technically one is bigger than the other, and they get the bigger piece, right? But they're sharing, right? But are they sharing equally? They're not sharing equally, okay? I don't know if that's a good example or if I have to come up with something better. But my sister always got the bigger piece. So polar covalent bond, everyone, in this example, we can see H2O, okay? H2O is what? Water. So we have 2 hydrogens and 1 oxygen, right? So remember, the hydrogens need 1 more to become full, each of the hydrogens, right? And then the oxygen needs 2, right, to also become full and stable, right? So because the oxygen needs 2 and each of the hydrogen needs 1, right, the oxygen is gonna share, and each of the hydrogen, they're gonna share their electrons with the hydrogen, right? As you can see here in this picture. But between the oxygen and the hydrogen, oxygen is more electronegative than hydrogen. And then that's the reason why when they're sharing these electrons, the electrons are going to spend more time towards the oxygen than the hydrogen, okay? They are sharing electrons, but the electrons are going to spend more time towards the oxygen than the hydrogen. That's why if you look at the second part of the image, you're going to see that we have this charge around the oxygen and around the hydrogens, right? It's showing that the oxygen is more negatively charged and then the hydrogens are more positively charged because the electrons are more spending time close to the oxygen, okay? So that's an example of a non-polar covalent bond. And then if they're sharing equally, then that's a polar, this one was an example of polar covalent bond, and if they're sharing it equally, right? If it's an equal sharing of electron, then that's an example of non-polar covalent bond, okay? Here in this image, we can see just water and then how water molecules and then how different oxygens attract different hydrogens, and then they make these bondings. This is just an example of ionic compound when we have a metal and non-metal come together to make an ion, just like this example here. This is an example of non-polar covalent bond. What did we say about non-polar? Do they share equally or not equally? Equally, right? Yes, equally. Non-polar covalent bond, and then you can see that they're all sharing equally. Here we have polar covalent bond, equal or not equal? Not equal, because here in this example, oxygen is very electronegative and sulfur is also electronegative, right? So you can see how we have this non-equal sharing of electron because one atom is more electronegative than the other one, okay? So everyone, do we understand, and then this is a table with the definitions, do we understand the difference between ionic bond, non-polar covalent bond, and polar covalent bond? Any questions? Because we're gonna skip to the next, jump to the next topic. Any questions about this? Okay, and then I think this should help you with completing the handout for the extra credit for Friday midnight. But still, if you are working on the handout and you're stuck on a question and you have any questions for me, please ask me and I will help you solve it, okay? Or any of the assignments, okay? Please ask me. And then please make sure that you turn it in, okay? Take advantage of all the extra credit opportunities. All right. Next, we're going to talk about different topics, and then those are some things that we need to cover. So everyone, what we're seeing here is enzyme-substrate interaction. Okay, last week, do you remember we talked about transcription, translation, how our genes, our genetic information needs to be transcribed to leave the nucleus, and then in the form of messenger RNA, it goes to the ribosomes where translation happens, right? And we talked about during translation how important these letters, these codons that code for anticodons are, because if there's a mutation, then it's going to code for the wrong amino acid. If it codes for the wrong amino acid, then we get the wrong protein, right? If you were absent last Wednesday, I recorded the lab. You can watch it in the lab 2 under the slides, okay? Because everything I said right now in like one breath is we spent hours talking about it last week, okay? But everyone, did you get confused or you were comfortable with this information about the proteins, protein synthesis? We're all comfortable with that, right? Okay, good. So we said that we don't want to have mutations because we don't want to code for the wrong amino acid, because one wrong amino acid can lead to the wrong structure of a protein, right? And then the structure of the protein is important because structure defines the function, right? The structure is very important, and then we're going to talk a lot about that every single week when we talk about different organs of different systems, because the structure is very important, and then things are structured that way to be able to serve their purpose, okay? So everyone, what you're seeing here in this image is the purple one is an enzyme. Enzymes are made up of proteins. Enzymes, all enzymes are made up of proteins. And then you can see that the enzyme has an active site. Do you see that? It's like a puzzle piece, right? And then that active site is very important because that active site is specifically for the substrate that's going to sit there, okay? And then this is the substrates that we have. Just pretend we have A plus B, and then we want to make the product AB, but we need the help of the enzyme to be able to do that, okay? So do you see the substrates here and the active side? In the second part of the image, you can see how the substrates, they fit in to the active side of the enzyme, right? And then in the third one is it's a transition state, so the enzyme, it's doing its magic. And then in the fourth image, you can see that A plus B is now AB, right? Two substrates, now we have a product, okay? Now, if the enzyme, the protein, did not have the right structure, right? Do you think the substrates were going to be able to sit on the active side? No. Okay. And then if that doesn't happen, right, then whatever this reaction is not going to happen. And sometimes these reactions are necessary to happen, then that person will have deficiency in whatever, whatever this reaction was for. Okay. So that's why the structure of the protein is very important. And then that's what we're covering in this slide, everyone. And we're also covering the job of an enzyme. Okay. And you see how like enzymes are important. We saw DNA polymerase last week for DNA replication. We saw RNA polymerase last week for transcription. We saw helicase enzyme, the one I said it looks like a scissor that unwinds the DNA double helix, right? So all of these enzymes are important. And all of these enzymes are made up of proteins. And then we need the structures to be correctly, right? Okay, any questions? All right, different topic. Here we're covering what are acids and what are bases. Okay, we have a pH, right? We have, what are the different pHs that we have, everyone? We have, what are the three pHs? Acidic, basic, neutral, and neutral, okay? That's correct. And then what is the definition of a pH? The definition of a pH is the concentration of hydrogen ions in a solution. It means how much hydrogen do we have in a solution? How much H plus do we have? The concentration of H plus in a solution. That is the definition of pH, okay? The concentration of H plus in a solution. So everyone here, you can see it in the beakers, right? For example, the top one says it's acidic, and then you'll see a lot of the H plus in the solution. The bottom one is more basic because you only see two H pluses in there, right? We see more OH minus, right? So you might be more familiar with this pH scale, right? So we have the pH, it goes from 0 up till 14, right? And then in this scale, we have like a range that the pH from this number to this number, it means it's acidic. We have not a big range, very small range for the neutral, and then another big range for the basic pH, right? So what you're seeing here in this picture, everyone, this first column, the dots that you see in the column, pretend these are the H plus ions, okay? And then you can see that at the bottom here, do you see how like we have more dots, right? So it means we have a lot of H plus. So it means that the lower we go down the scale of the pH, we get more what? Acidic, right? Acidic. So 0 is extremely acidic, right? And then acidic is usually up to 6, up to pH 6. It's going to be acidic, okay? And then the more we go higher in pH, it becomes less acidic, but it's still acidic, right? For example, the stomach acid, our stomach's pH is 2 or 3. It's very, very low pH. It's highly acidic, but again, why is it highly acidic? What happens in our stomach, which we're going to talk about today in the digestive system? Digestion, right? Breakdown of proteins. And then we need, so the stomach has a low pH, acidic pH, because of all the acids that we have in the stomach that are going to do digestion, right? So, for example, if we go a little bit higher, which is still highly acidic, we have the lemon juice, we have the vinegar. Vinegar and lemon juice are acidic. A lot of the times you see people using it as like cleaning tools, right? Because of how acidic they are. If you go a little bit higher, but we're still acidic, we have tomatoes and coffee, they're considered acidic. And then up till 6, it's considered acidic, but 6 is not as acidic as pH 1 or 2, for example, okay? Okay, what is pH 7? pH 7 is neutral, okay? So what is neutral? Water is neutral. Blood is neutral. Milk is neutral, pH 7, okay? And if we go higher, okay, now from pH over 7, right, 8 to 14, it's going to be basic, right? And then the higher we go, it's going to be more basic, right? Like 14 is more basic than 8, for example, right? And then you can see as we go up the scale, do you see that we have less dots, less H plus ions, right? Because it's becoming less hydrogen ions and more basic. And then what do we have basic? Baking soda is basic. Ammonia is basic. Bleach is highly basic, right? And then again, bleach is something that we use it as cleaning tools, and we're careful when we use bleach because it's highly basic, okay? Yeah. Anyone, any questions about the pH? Okay. Next, we're gonna cover, and then by the way, these are like small topics that you've seen here and there in Bio 5, right? Some are review, and then I'm adding a little bit information to it. So please feel free to ask me if you have any questions, okay? Next, we're gonna cover the 4 biological molecules, okay? And then after that, we're gonna start the digestive system. So what are the 4 biological molecules? Who remembers from last semester or when you took Bio 5, what are the 4 biological molecules? One of them is on the board on this slide. What are they? Carbohydrates, lipids, proteins, and nucleic acids. Good, good, good. Actually, that's the one that people forget, the nucleic acids, okay? So we have carbohydrates, proteins, lipids, and nucleic acids. And then we're going to review all these 4 biological molecules. Everyone, you've heard of all of this. We're just going to review it together. And then please let me know if you have any questions, okay? So first, we're going to start with carbohydrates, carbs, carbohydrates. So carbohydrates are made up of carbons and hydrogens, right? That's what makes a carbohydrate. And then we have a different way of drawing carbohydrates, like either C's with bunch of hydrogens surrounding it, right? Or we can draw these zigzags. Each angle is a carbon that's binding to 2 hydrogens, right? Or we can draw it as a ring, like the one at the bottom, or like a hexagon, right? Where we have each corner is a carbon and then it's binding to 2 hydrogens. So for carbohydrates, everyone, we have not just for carbohydrates, any of our biological molecules, we have something called monomers and polymers. What is the difference between monomers and polymers? Monomers is just pretend that we have one brick, right? One brick to build the wall. One brick by itself is a monomer because it's one brick, right? But if I take bunch of one bricks and I stack them on top of each other, I will build a wall of bricks, right? And then the brick is the monomer, the wall of bricks is the polymer, right? The polymer is made up of with bunch of monomers, okay? And then we have monomers and polymers for all the biological molecules. We have it for carbohydrates, right? We have it for proteins. Based on last week's lab, who can tell me what is the monomer for protein? If the protein is the polymer, right? What is the protein made up of? Bunch of monomers. What is the monomer for protein? Amino acids, right? Okay, good, good, good. That's good. So first, we're gonna talk about monomers for carbohydrates, and then carbohydrates are type of sugar, right? And then their names for monomer is monosaccharide. Okay, it means that one sugar. It's called monosaccharide. Saccharide means sugar. Mono means one. Monosaccharide means one sugar. Here in this example, everyone, we can see pentose sugar. It's a sugar with 5 carbons. and it's a monosaccharide because it's one sugar molecule, okay? We have deoxyribose, we have ribose, right? By the way, just by reading the names of these sugars, right? For example, deoxyribose, where have we heard that word before? DNA, right? Right? What does DNA stands for? DNA. Yes, deoxyribonucleic acid, right? And then what about RNA? What does RNA stands for? RNA, ribonucleic acid, okay? So everyone, I'm not sure if you remember from last week, remember the DNA double helix, right? And then remember we had like two strands, right, which makes the DNA double helix. And then the way that these double helix they are combined together is with their nitrogenous bases, right? The A binds to T, the G binds to C. These nitrogenous bases, right? They're complementary base pairs, right? But their complementary base pairs were changing, but the backbone, right, that makes the DNA double helix is the same on both sides, okay? On both sides, right before the nitrogenous bases, the A and T or G and C, we have a ribose sugar, okay? And or deoxyribose sugar, and we have a phosphate group, okay? So on both sides, we have, for the backbone of the DNA double helix, we have the phosphate group, we have the sugar, and we have the complementary base pairs, the nitrogenous bases, okay? And, oh, there's no image for that, but it's okay. So for DNA, the sugar for DNA is deoxyribose, okay? And then the sugar for RNA is ribose, okay? And so there's many differences between DNA and RNA. And I'm pretty sure you remember for Bio 5, on one of your exams, you were asked to list, I don't know, 3, 4 differences between DNA and RNA, right? And then you probably listed it because you memorized it. Oh, one is single-stranded, one is double-stranded, one has ribose sugars, one has deoxyribose sugars, right? But I want you to understand what's the difference between deoxyribose sugar and ribose sugar. They're both pentose sugar. They're made with 5 carbons. But if you look at this carbon here, this second carbon here, okay, the one that is on your lower right, okay, right before the carbon that has the COH and the H, right before that. So that carbon next to the H, do you see that it does not have oxygen? Right? And then, so deoxyribose, D means it does not, and then oxy means oxygen, and then ribose is just a pentose sugar, okay? So DNA has deoxyribose sugar, and then if you look at the other one, do you see that next to the H there is oxygen? Right? It has OH, right? So that one is ribose sugar. Okay, so that's the difference between deoxy means it doesn't have oxygen ribose sugar and then ribose sugar. So that's the difference when you look at the double-stranded DNA versus one-stranded RNA. RNA is made up of ribose sugar, whereas the DNA, right, it's made up of deoxyribose sugar. So it does not, it's missing that oxygen there, okay? Um, Here in this slide, we're just looking at monosaccharides of carbohydrates. So basically just simple, single sugar molecules. The most popular simple sugar molecule is what? Glucose, right? Glucose is like the most popular one, C6H12O6. We also have fructose, we have galactose. I'm not gonna ask you those, but I'm just introducing that these are monosaccharides, single sugar molecules. And there are different types of sugars, like glucose is the glucose that, whatever we eat, it eventually gets broken down into glucose, and then that glucose gets broken down into ATP. What was ATP? What do we use ATP for? Energy, right? So that's why glucose is like popular, right? The fructose, for example, fructose is a monosaccharide, simple sugar, but fructose is different than glucose because fructose is the sugar you get from fruits. It's different than glucose. We have galactose. So what I'm trying to say is these are just monosaccharides, single sugar molecules, okay? So now what do I do if I want to make a disaccharide? Disaccharide means it's a sugar that's made up of how many sugar molecules? Two sugar molecules, disaccharides, okay? So what do I do? For example, I want to build sucrose, right? As you can see here in this slide, I want to build sucrose. And to build sucrose, sucrose is made up of glucose plus fructose, okay? I have to combine these monosaccharides and make a disaccharides. Who remembers what is the name of the reaction called when you combine two monomers to make a polymer? What's the name of the reaction? I have two. I'm going to combine the two and then make one disaccharide. I have two monosaccharides. I want to make a disaccharide. What is the name of the reaction? What are we losing? Yes, the reaction is called dehydration reaction. And do you see where we're losing water, H2O, right? So dehydration means loss of water, right? When we're dehydrated, it means we're thirsty, we've lost water, right? And then water is, how do you write water? We write water as H2O, right? Two H's and oxygen. Or H and OH. H and OH is H2O, correct? Right? Okay. So here, if we look at the glucose, everyone, the glucose is going to lose the OH here, right? And then the fructose is going to lose the hydrogen. And then if I remove the OH and the H, what do I have left? I have the C from glucose, and then I have O and C from the fructose, right? So I will remove the H and the OH, and then whatever I have left, I'm going to combine them together, and then that's how I get sucrose. Did everybody follow? Right? So I just remove the H and the OH. That's called dehydration reaction. I'm losing water, and then whatever I have left, I just combine them together, and then now I have a sucrose, which is a disaccharide. It's a sugar made up of two sugar molecules. Okay? What is the opposite of dehydration called? Hydrolysis, right? Today Bio 5 said hydration, but it's hydrolysis. And then I wish it was hydration, right? Dehydration, the opposite is hydration. But the term is hydrolysis, because, sorry, I was going to use the term lysis. It's called hydrolysis because when we're doing the opposite, we're going to lyse. It means break the bond, right? We're going to break the bond and put the water molecules back, right? The H and the OH, we're going to put them back. To do that, we're going to lyse that bond, okay, and add the water back. So that's why it's called hydrolysis, okay? Hydrolysis means we're gonna lyse that bond for the disaccharide, add the water back, H and the OH. Now I have fructose and glucose, okay? And that's what happens during digestion, right? For example, you have a polysaccharide, right? You have to break it down into monosaccharides. And then you know how they say water helps with digestion, because every time we're breaking, right, a bond, right, we are adding what? Water. We're adding water. In this image, this is a polysaccharide. It's glycogen. Glycogen, everyone, this image here, each hexagon is one glucose molecule. And then here we have bunch of glucose molecules that are combined together to make a glycogen, right? And then you can see how each glucose molecule, how they're combined together. Each one has lost an H and OH, and then they're just holding hands, and they have a polysaccharide. Okay? Any questions about carbohydrates? All right, next we're gonna cover lipids. Everyone, did everybody understand the dehydration and the hydrolysis well? Because it's going to be the same for fatty acids. It's going to be the same for amino acids, right? For proteins. It's the same concept, okay? When you have two monomers, you want to combine them together. If you want to make a polymer, it has to go through dehydration reaction. They have to lose And then either side, it doesn't matter which side is losing the OH and which side is losing the H, okay? One loses the OH, one loses the H, or one loses the H, the other one loses the OH, and then we combine them together. And it's gonna be the same way for all the rest. So everyone, what we're seeing here is the second biological molecule that we're gonna cover today is lipids. And then everyone here, we can see the structure of fatty acids. So what you're seeing up there, right? You see a bunch of carbons and a lot of hydrogens, and then maybe like 2 oxygen, which is the carboxylic group, right? And then it's that one is labeled as saturated fatty acid. And then underneath that, right, we have again we have a bunch of carbon, but you'll see and hydrogens, but you'll see in the middle we have a double bond, right? And then it's called monosaturated fatty acid. And then all the way at the bottom, again, we have a bunch of carbons and bunch of hydrogens, but we have more double bonds, okay? Now, what is the why like why do I keep mentioning and talking about the double bonds? Have you like outside of biology or outside of class, have you ever heard about saturated fats versus unsaturated fats and how one is a better choice than the other, right? So which one is better for us, for our health, saturated fats or unsaturated fats? Yes, it's unsaturated fats, right? So unsaturated fat is better for us than the saturated fat. Now, where does the term saturated and unsaturated comes from? So I'm going to give you an example like from not biology example, right? For example, you know how they say it's not good to eat fast food, right? Because fast food restaurants they use the same oil, for example, all day for frying, and they don't change it as often, right? And then so the same oil, you're introducing it to heat several times a day, right? For example, if you cook it at home, you'll use it once or twice, and then you'll throw it away because it's for your family. But fast food chains, as long as there's the oil, it's frying, right? They keep heating up the same oil. So whenever we are introducing the oil to heat, what's happening is every time we heat it up, double bonds break and it adds hydrogen instead. Every time we heat it up, the double bonds break and we eat hydrogen. We add hydrogen instead. And then, so whenever you're heating up several times, right, that is getting more and more hydrogenated. It means we're breaking the double bonds and we're adding hydrogens, so it's getting hydrogenated. So whenever we say it's saturated, what is it saturated with? A lot of hydrogens, right? Because we're breaking double bonds. So that's what is not good for us, okay? Unsaturated fat, they are not as saturated with hydrogen. They have more double bonds, right? And then as long as it's not getting introduced to heat or it's not getting introduced to heat as often, right? Because whenever you do introduce it, it's going to get hydrogenated, okay? So that's the difference, everyone. The more double bonds it has, the less it is saturated with hydrogen. The less double bonds it has, it means that it's more saturated with hydrogen. Okay, so next time when you're thinking about, oh, is unsaturated better than saturated fat, just think of it saturated means saturated with hydrogen. It means it's hydrogenated. It means it has a lot of hydrogen, not a lot of double bonds. It means that that's not the good choice. The unsaturated is the better choice. Okay? Questions? Here, okay, this is the same example for dehydration or hydrolysis, but with lipids, okay? For example, we have a polymer for lipids is triglyceride, okay? And then we've heard of triglyceride, right? It's a form of a lipid. Whenever we're doing blood tests, part of our lipid panel has cholesterol, triglyceride, right? And all that stuff. But let's look at the structure of the triglyceride. Triglyceride is a polymer. It means that it's made up of with many fatty acids, okay? It's not one. It means that triglyceride is a brick of wall, right? And then it's made up of single bricks, correct? So how is triglyceride built? It's made up of glycerol, and tri means three, three fatty acids, okay? So here, everyone, we have a glycerol, and next to it, we have three fatty acids, right? So to make triglyceride, we have to connect each glycerol with a fatty acid. Do you see that? At the bottom, like the first line is the first fatty acid, the second fatty acid, and the third fatty acid, and then they're all combined to the glycerol, right? So for us to go from glycerol and three individual fatty acids through to triglyceride, how many oxygen molecules are, not oxygen, how many water H2O molecules are we losing because of dehydration reaction? Three, right? Because, so we're losing three waters because every time we're losing H from glycerol and OH from fatty acid, we're losing H2O, which is what? Water, okay? So, but that's how we build triglyceride with three dehydration reactions, right? We're losing H, OH, that's one, HNOH, a second one, and HNOH, the third one, and we have triglyceride. Any questions? Yes, three H2Os. And then did you understand why? Do you see how we have three fatty acids, right? Each one, do you see in blue, for example, the first line, glycerol H and OH, and then the second line, again, H and OH, and then the third one, again, H and OH, right? Each H and OH is one H2O. So we're losing three because we're trying to put together three fatty acids to one glycerol molecule. So we lose three water molecules. Questions? Okay, good. And then speaking of lipids, we talked about this last week, right? What is made up of lipid? Which part of the cell is made up of lipids? We talked about it last week. Phospholipids, bilayer. What is it? Which part of the cell? Plasma membrane, right? Remember the plasma membrane of the cell, which we said it's semi-permeable. It doesn't let just anything go in the cell or leave the cell. It's highly regulated. Sorry, it's highly regulated. And it's made up of phospholipid bilayer, right? Remember I was doing like this because we have the hydrophilic heads and the hydrophobic tails, right? And then the hydrophilic tails, hydrophobic tails are gonna be inside, and then the hydrophilic heads are gonna be towards outside, exposed, and they don't mind touching water, okay? Do you remember that, everyone? The hydrophilic heads, right? They like water. Hydrophobic tails, right? The fatty acids, they don't like water. That's why whenever we're drawing our plasma membrane, right? The phospholipid bilayer is built like this, right? The tails facing each other, and then the heads are outside or inside of the cell where they don't mind getting in contact with the aqueous solution, okay? Um, Here, this is just showing us how the structure of phospholipids. So we have the head part. The head part has nitrogen, carbon, oxygen, phosphate group. And then the tail part, we have fatty acids, two fatty acids molecule. And then one is unsaturated, one is saturated, because one side, it has more double bonds than the other side, okay? But that's too much details. Don't worry about it, okay? This is the phospholipid bilayer, and then this is the structure. We have other structures of lipids, such as steroids. Okay, in this image, we can see steroid and cholesterol. Those are all different types of lipids. And that's it for the lipids. Next, we're going to talk about proteins. Anyone, any questions about lipids? Are we all good? Are we all following? Okay. By the way, people in the back, I think there's enough seats in the front for all of you to come to the front, like one here, one there, and then two over there. Please, if you can come closer, okay? Now for proteins, okay? What are proteins made up of? amino acids, right? Proteins are made up of amino acids. Basically, the amino acids are the monomers, and then the proteins are the polymers, right? And then what we're seeing here in this image, everyone, this is the structure of an amino acid, okay? How many different amino acids do we have? 20. 20, we talked about it last week. We have 20 different amino acids. And then if you look at the structures of the amino acid, their molecular structure, how do we know that that structure is an amino acid? Because there are groups here that we're going to review, and all amino acids have these groups, okay? And then only one part of the amino acid changes, and then that's the part that differentiates between the different types of amino acids that we have, okay? So the most important part of the molecule that makes it an amino acid is this group here. It's called an amino group, okay? Amino group is NH2. NH2. What is NH2? What group is NH2? Amino group, okay? All amino acids, they have NH2, the amino group, okay? This is important. I don't know if ask this is anatomy. I ask this is in anatomy, but it definitely comes up in physiology. Okay, NH2 is the amino group for amino acids. Okay, all amino acids, they're gonna have that amino group. They're all gonna have that middle carbon. They're all gonna have that hydrogen at the bottom. And then on the other side, what is that other group called? Like based on the image? Carboxylic acid group, okay? Which is C double bond O OH, right? That is the carboxylic acid group. Um, all amino acids, everyone, they're going to have the amino group, that hydrogen at the bottom, and the carboxylic group, okay? And the only difference between the 20 different amino acids is gonna be the top group, which is, we've labeled it the R group. The R is just a letter. Instead of that R, we'll have like a different structure there, okay? And then on the next slide, I'm gonna show you different amino acids, different examples. And then we're going to see that all amino acids, they have the amino group, the hydrogen, the carboxylic acid group. And the only thing that is different from one amino acid to the other is which group? The R group, right? And then R group is, the R is just a letter. It's going to be like carbon, hydrogen, and other stuff there, right? But that's how we determine between the different types of amino acids. So everyone here, we have glycine, we have tryptophan, we have cysteine, right? So let's look at the glycine, everyone. Do we see the middle carbon? Do we see the amino group, the NH2? Do we see the hydrogen by itself at the bottom? Do we see the carboxylic acid group, the C double bond OOH, right? And then what's the R group for glycine? It's just an H. Glycine is like the easiest amino acid, okay? Next, we have tryptophan. Tryptophan, do we all see the middle carbon? The amino group NH2, the hydrogen at the bottom, the carboxylic acid group, the C double bond OOH, right? And then do you see the R group, right? So this R group, the green part, it should be green. It's green here. That part, everyone, is it's the R group that makes this amino acid tryptophan, okay? And then next we have the cysteine. Again, if you see that, can you tell that it's an amino acid? Because it has what groups? Amino group. What else? Carboxylic acid group, that H, and then it has a different R, right? We can tell that it's an amino acid. We might not be able to tell it's cysteine. Like I don't personally know all the amino acids and how do you draw them. But usually amino acids, whenever we need them, there's a table. We always follow the table. But it's important for us to know what is the basic structure of the amino acid, okay? Next we have, this slide, everyone. In this slide, we're just talking about, just like we talked about it in carbohydrates, in lipids, we're talking about dehydration and hydrolysis, right? So if I have two monomers, two amino acids, and then I want to join them together and make a dipeptide, what reaction do I do? Dehydration reaction, right? So everyone, do you remember in the amino acid, we have the carboxylic acid group and then the amino group, right? So we have the OH from the carboxylic acid group, and then we have hydrogen from the amino group on the other amino acid. Do you see that? The ones that are highlighted in blue, OH and H, right? This OH and H, it's going to be water, H2O. We're going to lose it, and then we're going to have what? A dipeptide, all right? This is the simply dehydration reaction, but we're doing it with amino acid, okay? You take the two monomers, one loses H, one loses OH, or vice versa, it doesn't matter. And like we can write it the other way around, glycine first, alanine first. It's exactly the same concept. We lose H2O, and then that's a dipeptide, okay? And then what's the opposite reaction called? Hydrolysis, right? Because we lyse that dipeptide bond, and then we put the H and the OHs back, and now we have alanine and glycine back, okay? Any questions? Okay, and then this slide, everyone is talking about the levels of protein. Oh, sorry, go ahead.
You said it doesn't matter where you take it out from, the OH and H?
So you'll put whichever's missing what, right? So you'll check the amino group. If the amino group is losing the H, then you'll put the H on the amino group. So for this one, you're gonna put the H on the amino group side for one of the amino acid, the OH on the carboxylic group side, yeah. Okay? All right, so next, everyone, any other questions? Right, and then I will give you a break shortly, okay? We're almost done with this part. Here we have the levels of protein structures. So everyone, we have primary, secondary, tertiary, and quaternary structure of proteins. And then in the next couple of slides, I'm going to magnify each one by one so we can talk about it. But basically, last week when we did translation, protein synthesis, and then we made that polypeptide chain, right? And then we said that, oh, that's going to be our protein, right? That's only the primary structure, right? The protein has to go through the secondary structure and the tertiary structure, and then finally, the quaternary structure that we see here, right? The last one, and then that would be a completed protein, okay? Do you all remember that slide that we saw the enzyme, right? The enzyme is made up of protein, and then did that enzyme look like primary, secondary, or tertiary structure, or it looked like the quaternary structure? It looked like the quaternary structure, right? I'm talking about this enzyme, right? This is a functional enzyme fully formed. So this is the quaternary structure, okay? But for the enzyme to get here, right? First, when we did protein synthesis or translation, right? This is how we started. Do you remember? Each tRNA was bringing an amino acid, right? Based on the codon with the anticodon and the amino acid it codes for. And then we ended up having a polypeptide chain of amino acids. And then we said that this is going to be the protein, right? So, and then everyone in this picture, do you see the part where it's magnified? Do we see like the NH2 and the C double bond OOH, right? Those are all bunch of different amino acid, and then they have different R groups, right? So now we can understand it if we see it. So that's the primary structure. The secondary structure of the protein is when the amino acids, they're already like holding hands with their polypeptide chain, right? With their peptide bonds, right? Here, when the amino acids, they interact via hydrogen bonds. They make hydrogen bonds with each other, okay? What we're seeing here, the secondary structure of the protein, everyone, it can be either beta pleated sheet or alpha pleated sheet, okay? Beta pleated means like this structure, like you're seeing here. And then amino acids, they can start making bonds with like one column with the other column, like with each other, not just the ones that they're connected to. The same for the alpha helix. Alpha helix, it looks a little bit different, but again, the amino acids are connected together, but they also make hydrogen bonds with the other amino acids, okay? Anyways, eventually they're just gonna like getting compact and coming together so that they take the quaternary structure, okay? And then this is the tertiary structure. We can see that now there's interaction between the R groups, right? And then they're coming together, it's getting folded, right? And then finally, this is the quaternary structure, which is the assembly of 2 or more polypeptide chains into the functional protein. Some protein might not need to go to quaternary structure to do their function, okay? But basically all proteins, they have this quaternary structure, and that's when they become a functional protein, okay? And then any questions about proteins? I was like, what were we talking about? Proteins? Okay, the last one is the nucleic acids, right? What are nucleic acids? What are our nucleic acids? DNA, RNA are what? Nucleic acids, okay? DNA, RNA are nucleic acids. And then everyone, oh, okay, this is the slide I was looking for. So everyone on the top, you can see you have a phosphate group. And then do you see the sugar, the pentose sugar? And then we have a nitrogenous base. Remember, we were talking about the DNA double helix, and then we were talking about, oh, the sugar for the DNA is the deoxyribose sugar, right? So the backbone of the DNA is going to have a phosphate group, the deoxyribose sugar, and a nitrogenous base on one side. And on the other side, again, it's going to have a phosphate group, the deoxyribose sugar, and a nitrogenous base that's complementary to the other side, okay? And then what are the different nitrogenous bases that we have? The purines and the pyrimidines that we were talking about it last week. What are the purines? Adenine A and guanine G. What are our pyrimidines? Cytosine C, thymine T, and uracil U, right? And then between DNA and RNA, we have thymine in DNA. We don't have uracil in DNA. And then we have uracil in RNA, but we don't have thymine in RNA, okay? Good. And then these purines, they are complementary bases with pyrimidines. A binds to T in DNA, A binds to U in RNA. G and C, they always bind to each other no matter if it's DNA or RNA, okay? Hmm? G, yeah,
Yeah.
G and C. G and C, yes. And then everyone here, this is the structure of nucleotides. This is the same image. We have a phosphate group. We have the deoxyribose. So the sugar and the phosphate group makes the backbone. And then the nitrogenous bases, it can be A, T, G, or C. And then this would be the one strand of the DNA, okay? And then these are our nucleotides, the purines and the pyrimidines. Another nucleotide, we have structure and formation of ATP. Everyone, what does the word ATP stands for? You can read adenosine triphosphate, right? Adenosine triphosphate. What is that? ATP. What is ATP? Is the energy, right? So we need energy, right? Our glucose molecules, they get broken down into ATP in several forms, and that's how we get our energy. And then everyone, for us to be able to spend the ATP, by the way, ATP, adenosine triphosphate, it means that it has how many phosphate groups? 3, right? Triphosphate. And then each phosphate group is PO4, okay? So here in yellow, do you see the phosphate group? 1, 2, and 3. Do you see the 3 Ps, right? With the PO4, PO4, PO4s, right? And then here we have a ribose sugar, okay? And then we have adenine up there, okay? So this is adenosine triphosphate. And then we need all 3 phosphates so that we can spend that energy. And then once we spend that energy, we lose a phosphate group. So triphosphate becomes, so whenever we spend energy, we need 3 phosphate groups. And then whenever we spend that energy, the 3 phosphate becomes 2, which is diphosphate, okay? So which is ADP instead of ATP. It means that we spent it. Now we need another phosphate so that we can have ATP again and spend that energy. Okay, this is what I wanted to show you. This is one of the strands, the backbone of the DNA. Everyone, do you see the P is the phosphate group? And then in the middle, the purple section, we have the deoxyribose sugar. And then here we have the nitrogenous bases, which can be A, T, G, or C. Okay, this is only on one side. So if you look at it here, this is the structure of nucleic acids of DNA and RNA. So everyone up here, you can see the DNA double helix and then the RNA, right? The DNA double helix is double-stranded, RNA is single-stranded. And then do you see the sugar phosphate backbone? So the part that is in purple, that's the sugar and the phosphate, okay? So you'll see here phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base. Here G is binding to C, okay? But what is the backbone? It's going to be deoxyribose sugar, right? And it's going to be phosphate, okay, on both sides. And then the same one for the one in the bottom. We have adenine and thymine, adenine and thymine. Again, you can see the phosphate, deoxyribose sugar, and then you can see the nitrogenous bases, okay? So everyone, now do you see exactly what is the molecular structure of the DNA molecule? The phosphates, the deoxyribose sugar, and the nitrogenous bases, right? But how do we draw it? We just draw like threads, right? Or X like a chromosome, right? But this is where it comes from, okay? The same for RNA, but RNA is, again, it has the sugar phosphate backbone. RNA also has the phosphate. It has ribose sugar, and it does have the nitrogenous bases, but it doesn't have T. It has U, and it's single-stranded, and the sugar is ribose. It's not deoxyribose. So these are just the nitrogenous bases. This is that same image magnified. You can see the sugar phosphate backbone, and in the middle, we can see the complementary base pairing of the nitrogenous bases. And then this is where that image is magnified from. If you look at the DNA double helix, and then this is the RNA. Everyone, this is a summary of our monosaccharides, disaccharides, polysaccharides, triglycerides, phospholipids. But if you understood the slides, you don't have to focus on these summary table. It's everything that we talked about, okay? All right, next we're gonna talk about digestive system. I'm gonna go ahead and give you like, just so that we have enough time, let's take about like 5 to 7 minutes break and then we'll get started, okay? Right now it's 3:30, let's be back by 3:37. All right, everyone, next we're gonna talk about the digestive system. And then I'll share with you in a little bit whatever I shared on the whiteboard when we get there, okay? We're not there yet. I just wanted to have it ready. So everyone, the first body system we're gonna talk about is the digestive system. The digestive system is very important, right? Basically, the digestive system is very important because all the food that we eat, it needs to get digested, right? So that we can get all the nutrients and then the blood can get the nutrients and then make sure that it spreads out, it distributes these nutrients to all the cells, to all the organs of the body, okay? So everyone, by looking at this image, can you name some of the organs of the digestive system that you see in this picture? And then digestive system is really not that hard. Like it's we don't add more organs than the ones that you learned in Bio 5, right? So it's one of the easier body systems because I'm pretty sure all of you are familiar, right? Or feel comfortable with some of the organs of the digestive system. Okay, so go ahead and name a couple. Small intestine, yes, yes. What else? Yes, liver, pancreas. Liver and pancreas are accessory organs, and we're gonna talk about that. What else? Large intestine, stomach, right? What else? Gallbladder, part of the liver. Someone said something else. Did I miss it? Esophagus, right? That's correct. The No, not cavities, organs. Yeah, organs. Pharynx, yes. Because like when we swallow it, it goes from the pharynx and then down the esophagus. Okay, so that's good. That's good. Anything in the mouth that helps with digestion? Tongue for swallowing, right? What else? Teeth, right? What do we do with our teeth? Chew, right? So digestion actually starts in the mouth, right? It starts in the mouth because, and it starts with mechanical digestion. Okay, what is mechanical digestion? When we're chewing, right, our food using our teeth, that is mechanical digestion. So mechanical digestion starts in the mouth with the teeth. And also, what else do we have in the mouth that does chemical digestion? Saliva, okay? So we're gonna learn, we have different salivary glands that produce the saliva that does the chemical digestion that we have in the mouth, right? And then, so then we swallow it, it goes down the pharynx, and then all the way down the esophagus, and then from the esophagus, it goes to stomach, that's correct. And then from the stomach, it goes next to the small intestine, and then large intestine, and then out, right? And like we said, we have some accessory digestive organs, such as the pancreas or the liver, with the help of the gallbladder, of course. And then we're going to talk about it and then see how they assist in digestion, okay? So, hey, everyone, you all know all the organs of the digestive system, okay? And then remember, for the lab exams, the questions are labeling the structures, the organs of the body systems. Digestive system is one of the easiest ones, right? You will get questions where you're labeling the liver, the esophagus, and stuff like that, okay? So good job on that. Here in this slide, everyone, we can see only the digestive system, like we've removed everything else. And then do you see how in the mouth, in the oral cavity, we have the salivary glands, right? Then we have the pharynx, esophagus, and then down we have the stomach. And then after the stomach, we have the small intestine. But do you see how the pancreas, right, and the liver and the gallbladder, we're going to learn today that we have some hepatic juices. Hepatic means liver, okay? Remember we said cardio means related to the heart, pleura means related to the lungs, right? When I use the term hepatic means that it's associated with the liver, okay? So we're going to have hepatic juices, means juices produced by the liver, and we're going to have pancreatic juices, digestive juices produced by the pancreas. And then you're going to see how these juices, both from the liver and from the pancreas, together, they're going to flow and enter the small intestine, okay? And then in the small intestine, I mean, the food is already going from the stomach to the small intestine, right? And then small intestine is where further digestion happens because we have the first, the small intestine is gonna do this mechanical peristaltic movement, right? So it's gonna keep digesting it. But further to that, we're gonna have the liver juices, the hepatic juices, and the pancreatic juices flowing the digestive juices in the small intestine, right? So further digestion happens in the small intestine. That's where you can see like there's a tube where it's merging from the pancreas and the liver to the route where we have the food's going from the stomach to the small intestine, correct? And then from the small intestine, it's gonna go to the large, then the next is large intestine and out, okay? The small intestine is basically the last destination where digestion happens and Also, what happens in the small intestine before we send the waste to the large intestine? Absorption, right? Good, good, good. So it's the last destination for digestion, and absorption takes place in the small intestine. Do we all remember last week when we were talking about the different types of cells and we talked about special cells? There was a cell that had these villi, right? What do we call those? Microvilli. Remember microvilli? We said the characteristic for microvilli was it creates large surface area for absorption, right? So if you look at these villi, which there's a slide, we're gonna see that, right? Each time that it's going up and down, it's just made up of bunch of cells, and then each cell has microvilli, and then the job of the cell is to absorb the nutrients, right? Because what's the purpose of eating, right? So everything is digested, we get the nutrients by the small intestine, it goes to the blood, and then the remaining waste or discard, it's going to travel through the large intestine and out the rectum, okay? So this is the big image of everything that we're going to talk about today, okay? Here in this image, everyone, we're seeing the abdominopelvic cavity. We've seen this in lab 1. Do you remember the peritoneum, right? The peritoneum is the organs that are found in the abdominopelvic cavity. Okay, we know that we have some organs in the abdomen, we have some organs in the pelvic area, and then we have some organs that starts in the abdomen and ends in the pelvic area, right? They intercept in both parts, and then basically the organs that are part of the peritoneum are the ones that are going to be protected by the serous membrane. Do we all remember the serous membrane? I was calling the serous membrane that double bubble wrap, right? Where we have two layers, the visceral layer, the serous fluid, and then the parietal layer, right? That double layer. The visceral layer was the layer that touches, gets in contact, direct contact with the organ. The parietal layer is the outside layer. Anyways, that's called the serous membrane, and then it protects all the organs. In this case, the peritoneum, which is the organs in the abdominal pelvic cavity, okay? And the opposite is retroperitoneal. Those are the organs that are not protected by the serous membrane. So this is just a review of that slide. Now in this slide, this is the information in this slide, and the next couple of slides is going to help us answer to the first question in our lab handout. What is the first question? What is the first question in the free response questions? Yeah, the organization of the peritoneum, right? So the organization of the peritoneum. So this is the answer to that question. It's written on the board. I'm gonna go over it one by one, okay? So actually, I also wanna try to do something here. Just because I wanna share it here. Okay. Mhm. This is for the recording. I wanted to share that so I can also talk about it. Okay, so this is the information that we're seeing here. It's the same information that I wrote on the board. Everyone, this is the answer to the first question about the organization of the peritoneum. So, if you want to take a picture, I'm gonna wait a couple of more seconds. And I'm gonna go back to my slides. Okay. So here, here, everyone, you can see that we said that we have the serous membrane, right? The serous membrane is that double layer membrane that covers the abdominal cavity, and then it's called the peritoneum that houses the organs in the abdominal pelvic cavity, right? And then we said that the peritoneum has two layers. The serous membrane has the visceral and the parietal layer, right? And then one is towards parietal is the outside lining, and then the visceral is the inside lining that it gets in direct contact with the organ. So these internal organs are protected by the visceral layer, which we call it the visceral peritoneum. So we're using the term visceral peritoneum or parietal peritoneum because these are for the organs in the abdominal pelvic region, right? For example, do you remember for the lungs, we said visceral pleura, parietal pleura, right? Or pericardium for the heart. So this is peritoneum because we're talking about the abdominal pelvic region. So everyone, we're going to review the primary folds for the peritoneum are first the mesentery, and we have the mesocolon, we have the greater omentum and lesser omentum. So before I go into details and explain each one by one, I want to tell you what are we talking about. These mesenteries, these mesocolons, right? What they are is they hold the organs in the abdominal pelvic cavity together. Okay, like have you ever wondered how like the liver stays here and then the stomach stays here and then the pancreas is behind the stomach, and then how the large intestine is placed here and the small intestine, and then how they stick and they stay in their place like the way that they are, the way that we see it, right? So there are some things that it connects it together, it makes it hold to the abdominal wall, or attach it to one another so that they can be placed the way that we see here in the model, right? So the first we're going to talk about the mesentery. The mesentery here you can see in the picture. Do you see that yellow part where you have where it's connected a little bit to the small intestine and a little bit to the large intestine, right? So the mesentery, the job is it connects the small intestine to the posterior abdominal wall. So that's how the small intestine is placed. It's connected to the abdominal wall, right? That's how we insert, like it's inserted, right? So that is by the mesentery, and then we can see the mesentery. We can see how the mesentery is connected to the small intestine, and then it connects the small intestine to the posterior abdominal wall. Next, we have mesocolon. Mesocolon is not in this image, but it's going to come up in the next slide or two. The mesocolon, everyone, it connects the large intestine to the posterior abdominal wall. Just like mesentery connects it to the small intestine to the posterior abdominal wall, the mesocolon connects the large intestine to the posterior abdominal wall, okay? And then you can remember that the mesocolon is for the large intestine because the large colon is the intestine, right? Next, we have the greater omentum. Everyone, in this image, the greater omentum that you're seeing, that yellow section at the top, that's not usually how it is. In this image, it's lifted up, okay? Another nickname for the greater omentum is the fat apron. So you know how like we place the apron, right? And then this is called the fat apron. And then here in this image, in this model, you're gonna get the chance to look at the models today. One side you can see the greater omentum, the other side is exposed, it's open. Or in this slide, you can see this yellow fat apron, right? That is going to be the greater omentum, okay? In this previous image, the greater omentum is lifted, right? Usually it's to the bottom and then it protects it, okay? So the greater omentum, basically it covers most of the viscera and is linked to the transverse column. So everyone, for our large intestine, the large intestine has ascending colon, the one that's going up, and then it has transverse colon like this, and then it has descending colon, right? The one that's going down, and then we have the sigmoid colon and then the rectum. We're gonna get there. But the reason why I covered this is because do you see how the greater omentum is covered to the transverse colon here, right? Is connected. So that's the greater omentum, okay? It's linked to the transverse colon. And the last one is the lesser omentum. Okay, lesser omentum, we can't see it in this picture because the greater omentum is covering it. Here you can see the lesser omentum. Sorry, lesser omentum. Do you all see this yellow part? This yellow part between the stomach and the liver? Yes? Okay, so this is how the lesser omentum, it helps the stomach to be suspended from the liver. Do you see how the stomach is suspended from the liver, how they're connected to each other, right? So these are the organizations of the peritoneum, the mesocolon, the mesentery, greater omentum, and the lesser omentum, okay? And you can see the greater omentum here. We can't see the mesocolon and the mesentery because the greater omentum is back to its natural state. We can see it only when it's lifted up, right? But here we can see the lesser omentum, how it connects the liver and it suspends the stomach from the liver. Okay, do we all see that? Okay, so this is the answer to question number 1. And then again, this is another image. Greater omentum is lifted. We can see how it's connected to the transverse colon. We have the mesocolon that attaches the large intestine to the abdominal wall. We have the mesentery, right? And here we've also labeled the different parts of the small intestine and different parts of the large intestine, which we're gonna get back to those shortly, okay? Where's my pictures? One second. All right. So everyone, next, any questions about the mesenteries and all that stuff? All right. Next, we're gonna talk about the basic tissue organization of the of most of the alimentary canal. Everyone, alimentary canal, basically, it's the whole passage where the food passes, starting from the mouth all the way till the anus. It includes everything, like from the esophagus all the way to the intestines and the stomach, right? And then just imagine like this long pipe or route where it enters from one side and then it leaves from the other side, okay? That's literally how our digestive system is. And then in this image, we're seeing how this pipe is made, okay? And then this whole route, this whole canal, and we're gonna see that it's made up of different layers, okay? Outermost layer, it's called the serosa, and then in other terms, serosa is the serous membrane. Serous membrane, what is the serous membrane? The visceral layer, the parietal layer, right? The membrane that protects it. So that outermost part is the serosa. Okay, this is like the whole canal, like you can see this in the esophagus, in the intestines, where the food is passing. After, like one layer inside the serosa is the muscularis externa, okay? It's the muscularis externa. And then the muscularis externa, it's all made up of muscles and it has different layers. For example, that outside layer is made up of longitudinal muscles. Longitudinal means like horizontal lines, like that's how the muscles look like. And then it's also made up of circular muscles, which is one layer more inside. You can see the muscles are circular muscles, okay? Circular layers. So serosa, muscularis externa, then we have submucosa, and then which is that white part, and then we have the mucosa, which is the inner layer, okay? So you can just imagine mucosa, submucosa, muscularis externa, and the serosa, but this is whole like one pipe made up of these layers, and then that's where the food travels from, okay? And then all the way inside, it's called the lumen. The lumen, that opening is called the lumen. And then that's like the inside where the food travels. That's where it gets in direct contact with the food. It's inside of the pipe, okay? Here we have oral cavity and pharynx. I'm not gonna pay too much attention on this one, but if you look at it here, we have an anterior view of the oral cavity. So we have the upper lip, we have the lower lip, we have the teeth, right? We have the soft palate up here. We have the uvula hanging here, right? And then I'm trying to cover the basic stuff first. The tongue, we said the teeth. On the side, we have the tonsils that we can see, right? And then we can see the tongue where we swallow. And then whenever we swallow, right? Remember, it's gonna go down the pharynx and then the esophagus. The pharynx, everyone, here, the pharynx is divided into three sections. So the pharynx is like one thing, but it has three sections. So the part where is like equal to the nasal side, nasal level, like the nose, it's called nasopharynx. And then the part where we swallow it, right? The oral part, the mouth part, it's called oropharynx. And then lower than that, it's called laryngopharynx, okay? Where we have the larynx. So everyone here, as you can see here in this picture, we have here, so the tongue, we swallow it, and then this is the pharynx, and then the pharynx has oropharynx here. Higher than here, it's gonna be the nasopharynx, okay? And then lower here, it's the laryngopharynx. So for the laryngopharynx, everyone, we have the larynx is the voice box, okay? And the larynx is not found on the esophagus. The larynx is found on the trachea, okay? What we're seeing here is two pipes, right? One is the trachea, where it goes to our lungs. That's the air pipe, right? And then the one behind it is the esophagus, which is the food pipe, right? And then, so the larynx is found here, but because this pharynx is almost the same level as the larynx, it's called laryngopharynx, okay? So nasopharynx, oropharynx, and laryngopharynx, but the whole thing is the pharynx, okay? And then the food's gonna go down the esophagus, okay? So here we swallow it, oropharynx, laryngopharynx, and then the esophagus. Remember that trachea is more anterior, esophagus is more posterior, right? It's two pipes, one for the air, one for the food, but one is front, one is in the back, okay? Now, both of these pipes are open, and that's a bit dangerous because, you know, sometimes, let's say when we're eating and we're talking, suddenly we start coughing and we say like the food went down the wrong pipe, right? So we need something that regulates it so that when we're eating, the food goes down the pharynx and then the esophagus. And then when we're not eating, when we're just breathing, right, the esophagus is closed and then the food is going, the air is going to the trachea, right, to the lungs. So what you're seeing here, is anyone familiar with the epiglottis? Where is it here? Okay, this white thing that we're seeing here, it's called the epiglottis. Epiglottis is this flap, okay? And then the job of the epiglottis is, it's a flap. It can either like cover the esophagus or it can cover the trachea, okay? Depends on what we're doing. For example, if we're eating, it covers the trachea, the esophagus is open, the food goes down the esophagus. If we are breathing, it covers the esophagus and then the food goes down the trachea, okay? And then sometimes, again, like I said, when we're talking and breathing at the same time and eating at the same time, sometimes the food goes down the wrong pipe and then we have to like cough it out, right? So is everyone clear on the function of the epiglottis? Yes, so it's a flap. It's going to close one of the pipes. This slide, let's go over it, but I'm not going to ask you to label the different teeth, tooth that we have. So in the front, everyone, we have the central canals, and then we have the lateral incisors, central incisors and lateral incisors, and then we have the canines, and then we have the premolars, and then we have the molars, okay? And basically, each teeth is designated to cut like different things, like molars are for herbs, canines are for meat, right? Structures have a function, and then it applies for the teeth. Here in this image, we can see a structure of a tooth. And everyone, you can see we have the root. The root part is the part that we can't see it. It's in the gums, right? And then we have the crown. The crown is the white part that we see it. And then also the animal. Animal is also the top part. And that's the part where, you know, they clean it. They see what's happening. Everything else, the neck and below the root is all gonna be hidden in the gums, okay? And then we have the dentin, dentin, and then inside of it, uh, we have, of course, you can tell because sometimes we bleed, right? We have blood vessels, arteries, and veins, and also we have sensory nerves, right? That's why sometimes we feel pain, right? And also we have the root canal, right? Which is also causes pain because we have sensory nerves over there. Okay, and that bottom is called the root, which is all the way down. It's going to be hidden in the gums. Next, this is important. I will ask you to label those. So remember, we said that in the mouth, chemical digestion starts in the mouth. And then during chemical digestion, we have the saliva in the mouth, right? That helps with digestion. So let's learn what are those three types of salivary glands that produce the saliva, where we can have chemical digestion in the mouth. So the first one is the parotid gland. It's all the way up here under our cheeks, you know. It's called the parotid gland. And then the next one is sublingual gland. Sublingual is under the tongue. And then that's how I remember it, because tongue, language, sublingual. Okay, so that's the sublingual gland. And then whatever we have our mandible bones, the mandible, the jaw here, right? The mandible. So over there, we have the submandibular gland. And then the submandibular gland also produces saliva. Okay, so can you name the three glands? What do we have? We have the parotid gland, sublingual gland, and the submandibular, okay? Do you think you can remember those? Okay, good, good, good. Next, everyone, this is the gross anatomy of the stomach here in this slide. So everyone, we swallowed the food, it went down the pharynx and then all the way down the esophagus. Now it's gonna enter from the esophagus to the stomach, okay? So this is the structure of the stomach. Everyone, the stomach is made from different sections, okay? And then to cover these sections, for example, as soon as it enters from the esophagus to the stomach, it's called cardia. And then on the side up here, it has the fundus. And then the rest, this whole thing is called the body. And then here, this last section is called the pylorus, okay? And then if it's a little bit confusing, this top cartoon diagram, it helps us understand that better. You can see right outside of the esophagus is the cardia. Up here we have the fundus. Majority of the stomach is the body, and then we have the pylorus. Then this is gonna be the small intestine, okay? So everyone, if you look at, you know, the fundus, the cardia, the body, let's look at the layers of the stomach. Stomach is made up of muscles, and then stomach needs to be very, very strong because it holds all the food. It holds the acids that do chemical digestion, right? So the stomach is a muscle, and then I don't know if you've heard they say that, you know, it can expand or it can get smaller because it's a muscle, right? For example, for example, for only like 1 or 2 days, if you eat a lot more than what you usually eat, your stomach is going to expand. Then you're going to have to start eating more so that you can fill up your stomach and feel full, right? I don't know if you've noticed, like by accident, if you haven't eaten like a day or so, I know it doesn't happen unless you're teaching all day and it's a Wednesday, right? your stomach starts getting smaller, so the next day you don't feel as hungry, okay? But once you start eating, then you start eating, okay? And then this is all because of the stomach is a muscle. It expands or it shrinks based on how we're eating, okay? I'm just trying to say that it's a muscle, and then it's a strong muscle. It's made up of the outer longitudinal layer, the middle circular layer, and then the inner oblique layer. So it's a muscle, and then do you see how it has like 3 layers of muscles, right? And then longitudinal, circular, and oblique, different directions. So it's very strong. Inside of the stomach, the body part of the stomach, it's called the rugae. This inside part is called the rugae, everyone. And then the last part, it's called pylorus. Okay? Now I want to focus on this area here between esophagus and the stomach. Okay, we have something like a rubber band, or I don't know, something that it does not allow the food, once it gets to the stomach, to go back up to the esophagus, because you don't want that to happen, right? What is the pH of the stomach? Highly acidic, right? You don't want, once the food enters the stomach and gets exposed to acid, travel back up to the esophagus. And then that's when we experience heartburn. Heartburn has nothing to do with the heart, but it's the acid, right? It's the acid reflux, right? When the food is going back up, and that's not good. That's why we have these sphincters, okay? Sphincters are there to control that some things in the body have only one-way passages, so it doesn't travel the other way around. So, and everyone, we have sphincters everywhere, and you're gonna see a couple of sphincters in the digestive system. The first one is here between the esophagus and the stomach, because we don't want the food from the stomach to go back up the esophagus. And the name is, you know how like the stomach, the digestive system, it's the GI tract, right? The gastro tract. Uh, and so the name of the sphincter, I'm just saying that so that we can remember it easier. Gastroesophageal sphincter. Gastroesophageal sphincter is the sphincter between the esophagus and the stomach, okay? And then, you know how we said this last part is called the pylorus? Over here, between the stomach and the small intestine, we have another sphincter there, because we don't want the food, once it enters the stomach, to go back up, once it enters the small intestine, to go back up to the stomach. So we have a sphincter there. And then because the last portion of the stomach is called pylorus, the sphincter is called pyloric sphincter. Okay, pyloric sphincter. It's a sphincter that doesn't let the food go back from the small intestine to the stomach. And yeah, and then what is the first portion of the small intestine called? Duodenum, right? And then the middle part, does anybody remember? I mean, we're gonna review it together, but just off of your head, do you remember what? Jejunum, yes, yes. And then the last one? I think you have to say it all together, and then the third one just comes up. Duodenum, jejunum, ileum. Okay? Like you just have to say that, and then the third one comes up. Okay? So the first part is the duodenum, and then everyone slowly we're gonna get to the part where first let me mention this. Do you see how we have the lesser omentum here, and then how the stomach is suspended from the liver? Right? And then, by the way, the liver doesn't look like this. You can barely see the gallbladder, but here in this picture, they've lifted the right lobe of the liver, so you can see more of the gallbladder. We can see more of the lesser omentum, how the stomach is suspended from the liver through the lesser omentum. So everyone, now that we covered the stomach, the pyloric sphincter, we're gonna get to the duodenum, which is the first section of the small intestine. And interesting things there is not only the food goes from the stomach to the small intestine, we're gonna talk about the different hepatic and pancreatic juices that are gonna dump their digestive juices in the duodenum, in the small intestine, where further digestion is gonna occur, okay? Any questions about this slide? Do we feel good about the stomach? Okay. This is the same image, but this is what a real cadaver image looks like, everyone. You see the liver, the lobes, the stomach, and then there's like a cross section of the stomach. The inside is called the rugae, and then it's showing us the pyloric sphincters, right? But probably the sphincters don't work as well because the person is dead. It's a cadaver picture, right? This is what a real picture looks like. And then, by the way, behind the stomach over there, that's the spleen. Do you see that? Here. Okay, that's the spleen. Also, to find the pancreas, if we remove the stomach, we'll see the pancreas right behind it. Okay? In this image, it's the structure and function of the stomach. So just like we talked about, you know, the different layer, the serosa, muscularis externa, submucosa, and mucosa. The stomach is part of the passage. It's made up of these layers. So we have the serosa all the way at the bottom. We have the muscularis externa, which is made up of longitudinal and circular muscles layers. And then we have the submucosa. And then submucosa is the layer where we have like the blood vessels. And then at the top, we have the mucosa. And then the inside is the lumen, or the inside part where the food is. It's the rugae part of the body of the stomach where the food is in there, right? It's getting churned. So on the top layer, we have mucus. And then everyone, if you look closely, right, on the mucosa layer of the stomach, we have cells called gastric glands, right? And the gastric glands, those are glands, they are on the lining of the stomach and they play a big role in digestion. And then all these glands, they have these mucus secreting cells. And then you can see all the way at the top, the mucus is lining the entire inside of the stomach. And then it's protecting the stomach lining from all the effects of the acids, hydrochloric acid, that is going to be released from these glands. And then these are the acids that do chemical digestion, okay? So we know that stomach is highly acidic. There's going to be acids there to do the chemical digestion. And then that's why we have the mucus lining to protect it from all the acids that the gastric gland is going to produce. All right, next we're going to talk about the small intestine. So, of course, this image is like magnified. This is not how our small intestine looks like, just so that we can cover the different parts. So what did we say? What is the first part called? The first part of the small intestine, the blue part. Duodenum, middle part. Jejunum, last part, ileum, okay? Duodenum, jejunum, ileum, okay? And then everyone, do you see the mesentery that connects the small intestine to the posterior abdominal wall? The yellow part, right? And then again, this is not what it really looks like, but the image is demonstrating it to us this way so that we can see everything, okay? So we have the duodenum, and then in the duodenum, that's where the food flows from the stomach to the duodenum. And shortly, we're going to talk about the liver juices and the pancreatic juices that also fall in the duodenum. Then they go to the jejunum and ileum, and then out the ileocecal valve where it joins the large intestine, okay? Any questions here? Okay, next slide. This is a cross section where we can see the inside of the jejunum, okay? Remember we said that small intestine is the last destination where further digestion happens and also absorption, correct? Right? So do you see these microvilli, right, going up and down, going up and down? And don't forget that these layers are the serosa, the muscularis externa, submucosa, and mucosa, right? It goes the same all the way till our intestine. Okay, and then inside we have the circular loads. I'm gonna focus here on these villi part. This is the intestinal wall. This is what really looks like under the microscope, and then that's a diagram. So everyone, do you see on the cells, okay, on the villi where it goes up and down, right? These cells, if you look at it closely, Okay, they have microvilli at the top because they're specialized cells. Okay, and then it's just a cell, just like any other cells, right? So these cells are lined up all the way through, every time that we go up and down. Okay, it's like all the way there. And these cells are called goblet cells. Okay, so these are the goblet cells. Their primary job is to absorb the nutrient when digestion is happening, and also goblet cells, they secrete mucus. Um, so this is it, everyone. This is the specialized cells that have microvilli, and they produce the large surface area for absorption. Next, we're going to talk about the large intestine. So everyone, here you can see after the duodenum, jejunum, ileum, right? The food is going to join the large intestine. This is where it joins the large intestine. That's where we have the ileocecal valve and the cecum. And then this is like a small pouch within this peritoneum, which is where we have the beginning of the large intestine. It's located on the right side. Okay, and then this is the appendix. When we hear like the appendix is bursting, that's the appendix. And then everyone, let's review together the parts of the large intestine. So this part that's going up, it's called the ascending colon. And then this part, the horizontal part, is the transverse colon. And the part that's going down is the descending colon. And then here we have the sigmoid column, and then out the rectum, okay? And you can see here the mesentery, right? The mesocolon. The mesocolon is the one that holds the large intestine, connects it to the posterior abdominal wall. Everyone, also on the large intestine, we can see these parts are called the diastrum. Let me see if there's anything else that I ask here. And then on the next slide, sorry, this is it. And then and then here we're gonna see that, remember we said we have sphincters everywhere, right? And then that's a good thing, right? Because we can control when we have to go for number 2 or not, right? Because of these sphincters. And also because we have sphincters, it means it's one way, right? So everyone, here you can see the large intestine. After the sigmoid colon, we have the rectum, the anal canal, and then down here we have external anal sphincter and internal anal sphincter, okay? And out the anus. So everyone, everything we talked about is the summary of the structure and function of the organs of the digestive system. The only thing left to talk about is the accessory organs, which is the liver and the pancreas. That's the only thing that we have left, okay? This is a summary table of everything we talked about. All right, next I'm gonna cover the pancreas and then the liver, and then we will be done. So here you can see the pancreas, everyone. For us to be able to see the pancreas, we have to remove the stomach. It's right behind the stomach, okay? And the pancreas is made up of the head, the body, the tail. It looks like this big leaf, right? And then the pancreas, if we look at the pancreas, everyone, here, right? These are the cells that make the pancreas, okay? The cells that make the pancreas, that yellow part, is made up of the pancreatic kidney, and it's made up of the pancreatic islet, okay? The pancreatic islet, so the pancreas, everyone, it's an organ that we see it here in the digestive system. It helps with digestion. We're also going to see it in the endocrine system, okay? And then we'll talk about that right now. And here we have the pancreatic kidney, okay? So the pancreatic kidney is the part of the cells of the pancreas that produces the pancreatic juices. The pancreatic islet has alpha and beta cells, okay? Where, let me start with the beta cells because it's easier. So what does the pancreas produce? What is it known for? Insulin, right? But more specifically, the beta cells of the pancreas, okay, are the ones that produce insulin whenever we have large concentration of glucose, okay? A lot of sugar, and then we need insulin to break it down. That's the job of the beta cells of the pancreas, okay? And then which pancreas, which part of the which cells of the pancreas? It's going to be in the pancreatic islet, okay? That's the beta cells. They produce insulin. The opposite is the alpha cells. Alpha cells, you know how, like, for example, sometimes when we eat too much, the body stores it so that whenever we are starving, we don't have immediate sugar, it goes to the storage and then it breaks it down and then it makes sugar and then we can use it again, right? So it's kind of like the opposite of the beta cells. Alpha cells in the pancreatic islet, they secrete the hormone glucagons whenever we have low concentration of glucose in our blood, so that now we have glucose again, okay? Beta cells, insulin, alpha cells secrete the hormone for glucagons whenever we don't have sugar in our system, okay? And then, like I said, the pancreatic kidney, that's the one that helps with the digestion. That's the part of the cells of the pancreas that makes the digestive juices, okay? And then everyone, the next thing that I want you to look at here is, do you see this line here? That's the main pancreatic duct. And then where is that going? To the duodenum of the small intestine, right? And then do you see where it's entering from, the pancreatic duct? There's an opening here, right? And then that's called the major duodenal papilla, okay? And then if you look at it here, it's not only the main pancreatic duct that is entering the duodenum, right? From the same opening, what else is coming here? The common bile duct, right? Remember we said that the liver juices, right, digestive juices, and the pancreatic juices, they're both gonna flow in the duodenum of the small intestine, right? So that these digestive juices help with digestion, correct? Okay, so are we good with the pancreas before we talk about the liver? Right? But I want you to see the bigger picture, that the same thing is gonna happen from the liver, and then it's from the same opening, and then it's going to enter here, okay? And I believe we have a sphincter here as well. We'll talk about it when we get there. All right, everyone, which organ is this? This is the liver. Actually, let's just talk about the liver a little bit. The top part is the anterior view of the liver. We have the right lobe and the left lobe. The right lobe is bigger than the left lobe, and then the gallbladder is found on the right or the left side? Right side, okay? And then we can barely see the gallbladder, everyone. Like, you can, I want you to take a look at the models once we're done, and I'm taking attendance. You can barely see in this model, you can barely see, like in green, the gallbladder. For us to be able to see the gallbladder, you have to remove the liver, turn it to the other side, and then look at on the right lobe, and then you can see the actual liver, which we can see it here, the gallbladder, right? Like this, if you turn it, you can see in green, the gallbladder, okay? And the gallbladder is very important, okay? Basically, what is the job of the gallbladder? Even though there are people that they've had gallstones, and they had to remove the gallbladder and they can live and survive without the gallbladder. But what is the function of the gallbladder? Basically, our liver makes bile. Bile is this fluid that it helps us digest lipids, fats, okay? The liver makes the bile and then it stores it in the gallbladder. So who makes the bile? Who stores the bile? The gallbladder, okay? So it's stored. It's made and released by the liver, and then it gets stored by the gallbladder. And then this bile helps with digestion. It breaks down fats. It breaks down fatty acids, right? That's why the people that don't have gallbladder, they recommend that they pay attention with the fats that they eat or the alcohol that they consume, because they don't have a gallbladder to store all that bile, okay? And then you're making it harder on your body because it doesn't have a gallbladder to store all the bile. Anyways, the liver has its hepatic juices. The right side of the liver has the right hepatic juices. The left side of the liver has the left hepatic juices, and plus we have the bile from the gallbladder. All of these juices, they get combined together, and then they leave through the common bile duct, right? And then remember where did the common bile duct leads to? To the major duodenal papilla, okay? So here we can see the front and the back, left lobe, right lobe, gallbladder, and then this is the common bile duct. This is the gallbladder. Um, what am I looking for? Okay, it's not in this picture. It is in this picture. Okay, so everyone here, the liver is lifted, right? We can see the gallbladder very easily. So the bile is coming from the gallbladder because it's stored there. Also here, do you see the right hepatic duct and the left hepatic duct, right? The juices from the right lobe of the liver, the juices from the left lobe of the liver. They're going to flow through the common hepatic duct, and then they're gonna join the duct from the bile. So they're called the common bile duct, and then they're gonna come here and then along with the pancreatic juices, and then they're gonna fall into the major duodenal papilla, okay? And then, like I said, we have a sphincter there, and it's called, the name is very easy because it's named after the liver and the pancreas. It's called the hepatopancreatic sphincter, okay? Hepatopancreatic sphincter. It's the sphincter that's found there so that the pancreatic juices and the liver juices, they don't flow back to the pancreas or the liver, right? Any questions about this? Did we understand the job of the liver and the pancreas, everyone? Questions? Okay, this is just like an supplemental image. We don't need it for anything. This is showing us a radiograph of gallstones in the gallbladder. This is a summary of pancreas, liver, teeth, tongue. Okay, all the tables that you have, like this is a big table. This is the same table, but broken down into two and magnified. And then this slide is just a summary slide of all the digestion that's happening, starting from the mouth all the way out, okay? Everyone, this is the end of lab 4. Please let me know if you have any questions.